Okay, good afternoon, everyone. I would like to invite Dr. Ravinder Agarwal, who is a professor in Department of Electrical and Instrumental Engineering at Thapar Institute of Engineering and Technology, Patiala. Welcome, sir. He earned his PhD in 1991 from Himachal Pradesh University, Shimla, and carried out his doctorate research at CSIR National Physical Laboratory, New Delhi. With over 3 decades of teaching and guiding 24 PhD students, Dr. Agarwal has authored about 170 journal papers, 250 conference publications, and 2 handbooks Springer published by Springer Nature. He served as one of the editors of the handbook of vibroacoustics, noise and harnesses, and as a sub-editor for 2 sections of handbook of metrology and application series from 2023 to 2025. Dr. Agarwal has successfully led several government-funded projects by DST, AICTE, ICMR, DRDO, and DITE. He is a fellow of IETE India, MSI, and USI India, and a life member of the Instrument Society of India. His research interests span around biomedical instrumentation, engineering sensors, soft computing, electronics, biological material characterization, and environmental monitoring. I invite Dr. Agarwal for the final defense of PhD Scholar Kale Sudarshan Nagorao. Welcome, sir. Sir, do we have your permission to start the one?
Yes.
Yes, thank you, ma'am. Thank you, sir. First of all, good afternoon to all of you. Myself, Sudarshan Kale. My topic name is Influence of Various Factors on Functional and Molecular Aspects of Semen Quality Associated with Male Infertility. I am doing PhD under the supervision of Dr. Ashish Agrawal, sir, and co-supervision of Dr. Jibanand Mishra, sir. So first of all, starting the introduction, I just want to take all of your attention on such a facts of infertility. So worldwide, 1 out of 6 couple is facing the infertility. Infertility affect around 50% of couple worldwide and 60 to 80 millions of couples universally. So TFR, that is total fertility rate, drastically declined from 1950 to 2100 from 7 child per female to less than 2 child per female in her lifestyle, in her whole lifespan. So this TFR for 2024 is estimated to around 2.03 for India. That should be 2.1, that is the replacement rate of the India. So TFR of Maharashtra, where this study is carried out, is 1.71. Again, it is the below level of the replacement. So male factor is estimated to contribute about 30 to 50% of cases of infertility. Drastic changes from 50 to 60% decline in sperm count from 1973 to 2008 as per the Lancet report, which was published in 2024. So in 2021, over 50% of the nations and their states had fertility rate that under the threshold for the replacement. So conclusion is that overall, if we are talking about the infertility, especially about the male infertility, sperm count, sperm motility or semen parameters are declining year by year. So infertility is the disease of male reproduction. It is failure to achieve the clinical pregnancy after 12 months or more of regular unprotected sexual intercourse. So female contribute about 40%, male contribute about 40%, and remaining 20% is the idiopathic or unexplained infertility, we can say. So there are the many causes of infertility. It may be the genetic factor, it may be the changing lifestyle, alcohol, tobacco, drugs, medication, poor diets, environmental influences, etc. All these factors can affect the semen quality and convert normozoospermic sample to the azoospermic, teratozoospermic, oligozoospermic, asthenozoospermic, or combination of the oligo, astheno, terato likewise. So azoospermic means no spermatozoa in semen sample, terato means no, terato means abnormal morphology, oligo means low count, astheno means having the less motility, etc. So this is a diagram showing the 40% male factor, 60% other, 40% is female, and 20% is unexplained infertility, that is unknown factor. So this is a concern as it is a social concern rising in India as well as universally. So there are some factors like socio-demographic factor, environmental lifestyle choices, biological and genetic causes. All these factors are affect the semen quality. There may be the genetic risk which impact the semen quality parameter, fertilization failure. So if we are talking about the male infertility, so we start the assessment of male infertility beginning with the medical history questionnaire and then semen analysis. Inside the semen analysis, physical parameter like volume, viscosity, color, pH, and microscopic parameter like concentration, motility, morphology, viability, DNA fragmentation, that is DFI index, etc. we are going to analyze. So these all are these factors generally affect the spermatogenesis or spermiogenesis. There may be a mechanical obstruction as well. So these are the factors. If you are talking about the semen, this is the composition of semen. As we can see, 15% fructose, 20% protein, enzymes, hormone, mineral, only 5% spermatozoa, means sperms are present inside the semen. So these are the semen parameter. First of all, these are the major semen parameters: sperm concentration, number of sperm, sperm motility, that is progressive, improgressive, non-progressive, then sperm morphology, that is shape of the sperm, semen volume, pH level, and WBC, RBC, all are the present in that semen. Okay, so as per the WHO criteria, this is the referral value provided by the WHO to analyze the semen sample. So as per the WHO, sperm count should be 15 million per ml. Then progressive motility should be 32%, whole motility should be more than 40%, morphology should be more than 4%, semen vitality, live sperm should be 50 more than 58%. This is the reference value provided by WHO, that is World Health Organization. And by using this parameter, we give the impression to that sample. If there is a less concentration of sperm, then it is a oligozoospermic sample likewise. Okay, this is the major and key contributing factor behind the male infertility. First of all, sperm disorder like low sperm, no sperm, poor sperm motility, etc. Second one is the hormonal imbalance, low testosterone, imbalance in FSH, LH or other hormones. Physical obstruction may be there, that is blockage of the vessels differentia, ejaculate duct, or there may be a varicocele. Genetic factor like Klinefelter syndrome, Y chromosome deletion. Lifestyle factors, smoking, alcohol, drug, obesity, exposure to the toxin, medical, if there is a previous infection, cancer, diabetes, thyroid, these all are the including medical condition. There may be a cultural stigma and lack of awareness around the male reproductive health, and late marriages and delay conception. These all factors contribute to the male infertility. Okay, if we are talking about the global trends in infertility, yeah, there are the delay childbearing, lifestyle factor, environment factor, and medical conditions. These all are the factors which contribute the universal infertility, and this is the current status of infertility globally. As I already told, TFR is continuously declining, approximately 5.0 in 1950 to 2.2 in 2021, and this is again further projected to decline to the 1.83 by 2050 and 1.59 by 2100. So again it is a below the replacement level. Yeah, there is one sub-Saharan African, they have the right now higher TFR than the other countries. There may be a policy implication like Hum Do Hamare Do likewise policy, they may responsible for the decline in TFR globally. Okay, this is the recent 2024 reports of Lancet. They said that it is declined from 5 to 1.59. Okay, this is about the infertility in India. So as I already said, there are some factors like cultural shift, that is where moving to the small family, health issues like hypertension, diabetes, all are increasing in India. Pollution, air pollution, water pollution, etc. factors are responsible for, and in again there are in rural area there is a no facility of healthcare, so they are unaware or leading to untreated infertility issues. So these factors are responsible for the infertility in India. If I am talking about the current status of infertility in India, that is, as I already told, TFR rate of India is below the 2.1. So current TFR rate for India is 2.0. And if you see this diagram, there are only 4 to 5 states like UP, Bihar, Meghalaya, Jharkhand, Madhya Pradesh and Rajasthan. These 5 states have little bit more TFR, that is more than 2.0. But remaining all states have the less than TFR is below the 2.0. So it is a total fertility rate, we can say, declining. So in Maharashtra also it is 1.7, even for our Punjab it is 1.6. Okay, so as fertility problem is getting increased, so peoples are trying to conceive artificially by using the treatment, medical treatment, and that treatment we call it as a IVF industry or in vitro fertilization. So if we can see in 2015 that industry market approximately about 16.89 billion. In 2023 it was 25.3 billion, and in 2025 it is 32.16 billion USD dollar. So we can say that within the 10 years it almost 100% increasing the growth rate of the IVF industries. So there are many IVF chains in India like Nova IVF, Indra IVF, Nilkant, Oasis, etc. So they are currently growing faster. So I go through all this review literature. They suggest that yes, there is the impact of age, BMI, environmental on the semen quality. Yeah, this is the research gap we found when we started our research. So in India there is study of correlation of multiple factors on semen quality were missing on same population, that is age, BMI, lifestyle, seasonal. Limited study with less information only available about the association of changed lifestyle and male infertility. So seasonal variation and their impact also not that much revealed, and correlation of various factors and their relation with DNA fragmentation is where missing. So as per our knowledge, our studies only study from Maharashtra. These are our objective. First of all, semen collection, storage along with the personal questionnaire. Then we will evaluate the functional aspects of semen quality, then molecular aspects of that collected semen sample, and find the impact of age, BMI, lifestyle factors on semen parameter, that is functional as well as on molecular level. So this is the block diagram of our study. Here we can see subjects were visited to our clinic, then questionnaire collected, BMI calculated, inclusive and exclusive criteria are there. So those are within the inclusive criteria we collected semen sample for those subjects. So first of all we carried out the functional test, that is semen volume, sperm count, motility, morphology, and other one is the undergoes the molecular testing, that is DFI by SCD method. So for calculation of the functional test, we again collected that data and stored that data in Excel form. We categorize the data as per our factors. That first factor is impact of health factor, second one is the lifestyle factors, third one is the effect of diet, and fourth one is the impact of sexual abstinence, last one is the environmental impact. So again this main factor having the subheading, like if you are talking about the impact of health factor, it means age, BMI, medical histories. Then if you are talking of the lifestyle, cigarette, smoking, tobacco, these are the subcategories we are included in our study. After that, after collection of all data, we we did the statistical analysis, correlation, technical parameter perform, and finally we got the results and conclusion. So as I already said, this is overview of our study. We have collected semen sample, analyzed and stored the data as per our categorized. Okay, for doing this research we require first of all ART lab, that is assisted reproductive technology lab, which should be equipped with the all necessary instrument like laminar earflow, microscope, centrifuge, pipette, macular chamber, etc. Then we require semen sample, then we require DFI kit, then SPCS software for the analysis, statistical analysis. Sorry. This is the sampling procedure. As per the inclusion and exclusion criteria, we have collected the semen sample. Sample age is in between the 18 to 55 years. A place of data collection in FemCare Fertility Center, Pune, Maharashtra. Okay, this is a grouping of all factors. As I already told, there are many factors. So if you are talking about the age, we categorize age into total 6 factors, that is less than 25, 26 to 30, 31 to 35, 36 to 40, 41 to 45, and more than 45. Same we are did with the obesity factor, that is underweight, normal, overweight, obese, and obese patient. So as like the smoking habit, consumption of tobacco, alcohol intake, type of diet, that is vegetarian or non-vegetarian, medical history having the past medical history or not, type of drinking water, that is filter water using or non-filter, consumption of fast food, food supplement, tea, coffee, fizzy, that is carbonated drink. So these are the grouping we first of all did. After that, this is our 4 methodology we are used for achieving our objectives. Thank you. First of all, assessment of the impact of factor on functional parameter. It means these all factor, all previously mentioned factor, how they impact on count, motility, morphology, that is a functional parameter. Second was the evaluation of influence of this factor on DNA fragmentation by using the sperm chromatin dispersion method, that is SED method. It is for molecular analysis. Third one is the impact of sexual abstinence period, that is SAP, on semen quality. And last one is the how seasons influence the semen parameters. So these are our 4 methodology. We will see one by one. So this is the first methodology: questions are collected, semen collection, macroanalysis, microanalysis, and their impression. So these are the tools and equipments we are used. That is, this is a semen collection container, name, date, abstinence period, etc. This is a andrology lab at right side. This is a macular chamber for sperm count and motility, centrifuge machine, and this is a for morphological slide. Next one is the evaluation of influence factor for DNA fragment, that is SCD method. Same for collected semen sample, we use this kit. So this kit provides the total 4 solution ABCD. First of all, we have to melt the agarose, mixing of the agarose in that semen sample, slide preparation. Sorry. Placing cover slip. Then for solidifying we use the 4-degree temperature, and this is what we get after the results. So sorry. Thank you. Okay. This diagram shows the there are 4 types of spermatozoa here after staining. At this right side, we can see at the corner of right side, that is a sperm which having no halo. Then in middle there are large halo, medium halo, and at the below corner there is a very small halo. So those having large and medium halo are the normal spermatozoa. Those are the no halo and small halo are the fragmented spermatozoa. So more precisely, this is the diagram, that is halo analysis. Okay, our third methodology is for sexual abstinence and their impact on functional as well as molecular aspects of the semen quality. So here again we group the data as per the group first 0 days abstinence, group second 1 to 10 days, 10 to 20 days, and more than 20 days. So we get then we categorize the data as per the test performed, and last one is the seasonal impacts. So we again group that seasons in our as per our meteorological department: summer, monsoon, autumn, and winter, that is January to February. So this is the diagram for season. So as per methodology, we have we got the results and we have arranged the results as per our 4 used methodology. So we will see one by one. Is first of all assessment of the impact factor on their functional parameter. So in this parameter, first factor is the age. So how age impact the sperm count, motility, morphology. So this is the first diagram which are showing the impact of age on sperm count. So here we can see as age increase, sperm count also get increased. So 20-25 years having less sperm count, and it is get increased by age. This is our second diagram which are showing the semen volume and impact of age on semen volume. So here we can see semen volume remain relatively stable in between the 26 and 45 years, while before and after this age group it was shown slightly decline. Next one is the progressive motility. So progressive motility we observe in the age group of less than 25 years is quite highest than the other age group. This is a combined, we can say, impact of age on semen quality considering each factor. Okay, next factor is the BMI. So there are the total 4 different categories of BMI, as already said: underweight, normal, overweight and obese. So we can see sperm count we were observed in the normal weight of the BMI. Then impact of BMI and semen volume. Here we can say decline volume of the semen observed in the obese group. Next one is the BMI on total motility. So there are different 3 types of motility of spermatozoa: total motility, progressive motility and non-progressive. So here we are use the total motility. So total motility we found in normal weight of the BMI, which is noted higher. Same about the progressive motility. So normal weight BMI group patients show the highest average value of the progressive motility, that is 24.87%. Obese group showing the again declining progressive motility, which is very important parameter of the semen. This is the sperm morphology. We can see 1.74. It is normal weight BMI group patients show the highest average value of the sperm morphology, 1.74%. And this is a combined effect. So we can see that very less combined effect were found in the normal weight. Combined effect means, for example, there is a sample which having the normozoospermia, there is a 0 combined effect. Asthenozoospermia, there is a 1 defect. Asthenoteratoid, that is 2 combined effect. So we can see that normal having the very less combined effect. So next parameter is the smoking. So here we can see that there are different types of the group, that is 0 time smoker, 1 to 5 time smoker, 6 to 10 time per day smoker, more than 10 times smoker. So 32%, that is those who are 0 time smoker, we found the 32 million per ml count. Then impact of the smoking frequency, that is 2.1, we found that volume again found in higher in 2.0 times. Smoking frequency and total motility, yes, here we unexpectedly found that more than 10 times smoker having the higher total motility. Same about the progressive motility. Then impact of the smoking frequency on morphology. So in that case, we again unexpectedly found that more than 10 times having the higher morphology. So this is the smoker and non-smoker yes or no category table. Those are the non-smokers showing the higher volume. Those are the non-smoker showing the higher sperm count. Again, non-smoker showing the highest total motility, progressive motility, and as well morphology as well. So this is about the smoker and non-smoker. Then this is the impact of tobacco consumption. So here we can see impact of tobacco consumption on sperm count. So unexpectedly we found that sperm count were noted highest in the group of subjects who consuming tobacco 1 to 5 times per day. So and in remaining groups it is stable. Same thing we are observed about the semen volume. So total motility of sperm were noted lowest in the group of subject consume more than 10 times of the tobacco consumption per day. Next one is the tobacco consumption on progressive motility, where was highest in the group subjects consuming tobacco 1 to 5 times per day. This is a morphology. We found that 1 to 5 times tobacco consumer having the little bit higher morphology, 1.81%. So 0 times sperm consumer having the combined effect quietly less. This is a yes or no table. So those who are saying no to those who are non-consumer having those who are consumer having the highest number of sperm count, total motility, progressive motility, morphology, and even they have the higher combination of the combined effect. The impact of alcohol consumption, those who are consuming alcohol having the highest sperm count, highest total motility, progressive motility, but less morphology and highest combined effect. This is the impact of diet on vegetarian on semen quality. Here we can clearly see that those who have the vegetarian diet, they are having the higher sperm count, having higher total motility, progressive motility, morphology, as well as having the higher combined defect. So we can say yes, vegetarian diet is good for the semen quality. Impact of medical history, we didn't found any major difference in the group, both group. Yeah, filter and non-filter water, how they affect the semen quality. So those subjects are taking water from water filter, having the good sperm count, having the good total motility, progressive motility. This is the impact of fast food, that is burger, vada pav, and all the semen quality. So those who are non taking the fast food, having the positive impact on their semen quality, that is count, morphology, total motility, progressive motility. This is the impact of food supplement. Those who are not taking the food supplement, having the good semen count, motility, morphology, progressive motility. This is the impact of tea coffee consumption. So we can say that those who are taking the tea coffee, having the good or positive impact on semen quality. This is a impact of tea coffee consumption group, that is those who are taking 3 to 4 times per day tea. So they are showing little bit positive impact on their semen outcome. So this is the impact of carbonated drink, that is Coca-Cola and all. So those are not taking the consumption of this fizzy drink, having the good sperm count, motility, morphology. The impact of combined caffeine. So those who are taking the caffeine, slightly having the positive impact on their semen parameter. This is the impact of unexceptionally, we found that the impact of pesticide and chemicals. Those who are having the exposure to the chemicals and pesticide, they are little bit showing the higher value of the sperm count, motility, and morphology. So this is the heat map of multivariate coefficient. So the red star mark show the significant relationship among the variables. So here that red means BMI is directly affecting the DFI. So likewise that. So this is about our first methodology, how this factor affect the functional parameter of the semen. So now is the time to evaluate the impact of this factor on the DFI or DNA fragmentation by using the STD method. So this is the norms which are provided by WHO. So if DNA fragmentation is less than 50%, so it is a good sperm. 15 to 30 is that is a very good sperm. This is a 15 to 30 is a good sperm, and if fragmentation is more than 30, then it is a abnormal sperms. So first we will see the how age affect the DNA fragmentation. So here we can clearly see that those having the age group of 21 to 25 having the less DNA fragmentation. So age we can say directly affect the fragmentation. Second one is the BMI. Here is again BMI is increasing. So those who having the normal weight are having the very less DNA fragmentation, even though underweight, overweight, or obese patient having the highest DNA fragmentation. So BMI is very important factor which directly affect the DNA fragmentation. Next one is the lifestyle factor. First factor is smoking. So those are smoker having the highest DNA fragmentation, that is 36.29%. So this is the again categorized. Those are taking 6 to 10 cigarette per day having the highest DNA fragmentation. This is the impact of tobacco consumption. Yes, those who are consuming the tobacco having the highest DNA fragmentation, that is 33.06%. This is again those who are taking more than 10 times consumption per day, which are showing the very highest or drastically highest DNA fragmentation than the other comparing group. This is the tobacco consumption percent. Those who are tobacco consumption, I already say that this those who are taking more than 10% having the highest. Okay, this is the tobacco consumption and smoking. Those person who consume both tobacco as well as smoking. So those who are saying yes, having the 41.83% of fragmentation, whereas those saying no, they just have the 34%. It means it is a combinedly impact the DNA fragmentation. This is the alcohol consumption on DNA fragmentation. So we didn't find that much variation of alcohol affect the DFI value. This is a past medical history. Again, we didn't find that much variation in the medical history. Again, the DNA fragmentation. This is impact of diet, those who are taking veg or non-veg diet. So both group having almost same DNA fragmentation. Filter of water filter, so doesn't, here again we doesn't get the significant effect of the DFI value on semen quality. DFI percentage, again the fast food, so little bit here we found the slightly higher, those who are not consuming the fast food having the lesser DNA fragmentation. Then those who are taking the food supplement, which those who are taking the food supplement having the highest DNA fragmentation, here we found that, so which suggests higher presence of the abnormal spermatozoa. Tea coffee consumption, we didn't find the major difference, no significant difference in DFI groups in the both. Those who are taking 6 to 10 times of the tea per day, so they are noted highest DNA fragmentation. As though this is a carbonated drink on the we didn't get that much conclusive difference as like the chemical exposure as well. So we can see this is our third methodology, impact of the sexual abstinence. Sexual abstinence period means there is a subsequent gap between the two sexual intercourse. So here we have categorized this data in for analysis of the first of all functional parameter, and second one is the molecular parameter, that is DFI. So this is about the first parameter, that is functional parameter, how sexual abstinence period affect the functional parameters of the semen. So here we can see that there are total 4 types of group: 0 abstinence, 1 to 5 days abstinence, 6 to 10 days abstinence, and more than 10 days abstinence. So those patient or those couple having the abstinence period of 6 to 10 days, which are showing highest volume, highest sperm count, highest morphology, and lesser combined defect. But yes, as I already told, motility is a very important parameter of the DFI. So those couple who are having the lesser abstinence period, that is 0 days, are showing the highest total motility and progressive motility of that semen sample. So this is the impact of sexual abstinence period on DFI. So we can consider 6 to 10 days of sexual abstinence having the additional benefit among the group of showing the lowest DNA fragmentation, that is 31.83%. And our last parameter or last methodology in how seasons are affect the semen parameter. So this is the table which is suggesting, or where we can see that summer, monsoon, autumn, and winter. So in winter, sperm count, sperm total motility, sperm progressive motility, even though morphology, all we are found highest in the winter season. This is the same where here multiple line graph showing the winter having the highest peak. This is the violin plot. This representing the normal morphology for different seasons. So here we can see in winter, outward 2 bubbles are there, are having the larger size. They are denoting that yes, morphology are noted in higher in the winter season. This is a swarm plot which are representing the motility for different seasons. So here we can see in the winter season that pink dots are higher in above, that is more than 30% or 40% compared to the summer, monsoon, and autumn. So this is about the discussion. There are very previously study also suggested that the impact of the these factors on the semen parameter. But as our study are showing the how this factor combinedly affect the semen quality. So among the parameter used, evaluate the condition of patient in relation the different lifestyle factor considered in presenter work. So it can clearly observe that younger age, normal BMI, non-smoking, less frequency of tobacco consumption, and less frequency of alcohol consumption is the most effective indicator for their effects. So this is summary. We can say if you are talking about the age, sperm count increase with age. Motility is highest in less than 25 years group. Semen volume is stable. Then morphology is unaffected by age. then younger group has the less DNA fragmentation. If I am talking about the BMI, normal BMI is good for all semen parameters. Smoking or reduce the sperm count and volume. Tobacco 1 to 5 day per is showing the best count motility, but it may be a temporary effect. Then for alcohol we didn't found the major difference in semen parameter, but yes, DNA fragmentation found higher in alcohol consumer. Diet, vegetarian diet is good for the health. Cold drink, food supplement should be avoided. Fast food and supplement may link to the increase in DNA fragmentation. Tea, coffee show the slight positive effect on semen parameter, so higher consumption may increase the DFI slightly. So pesticide chemical exposure should be avoided. Sexual option 6 to 10 days should be the good for semen count, semen quality, and winter season is good for the conceiving naturally. So this is again conclusion that I already say, but yes, expert agreed that both partners should be checked when they are dealing with the infertility. Sperm DNA quality is one of the best indicator for male infertility. Since many factors are involved, we need to more large and detailed study to find the clear answer to better way to help the couple facing the fertility problem. So for understanding the this infertility overall, so we may need the interdisciplinary approach. We may need the multiple demographic, cultural, geographical samples to clarify the significance of their impact on semen quality. So this is our publications. This is a list of conferences. Thank you, sir. If we are talking about the DNA fragmentation, we found there those who are chewing the tobacco having the higher DFI. So there are maybe DNA fragmentation increase in those patients.
And in some part of the country you are seeing it's a problem.
Yes, sir.
In hilly areas.
Actually, this study is carried in Pune, Maharashtra only. But as if we are talking about, as always, that diagram is showing 3 to 4 states only having the higher TFR. Remaining all the states having the low TFR. They have the fertility issues.
What are the major factors there for this?
Actually, major factors, if in my view, if you are talking about the lifestyle is the major factor in all over the state, even in universal, this is the major factor we can say. Remaining, that is maybe medical services, if you are talking about the hilly areas, cold. So as our body, like scrotum and all, they are working, our body is thermostable, we can say. If higher cold is there, so it may affect the sperm count motility in the hilly areas. So their thermal temperature may not be that much, so that may be the reason.
What are the problems you have faced during your research work?
First major problem is I have faced is the sample collection, sir. So this topic is very personal. If I am talking about the sample collection, first of all, no center is ready to give their personal data to us. So why they will share us? So initially, and then meanwhile COVID. So 3 years of COVID are very tough for us. So we have already collected sample in one of the center. After COVID then whole 3 years we just vanish it off. Again, we freshly start up with one of the IVF center.
I think
So they give the consent.
that IVF center are the best center to collect the
Oh, sorry, sir.
sample. IVF centers.
Which IVF center is best? Okay.
Good number of centers are available.
There are many IVF chains, I already said. If we are talking about the IVF industry, Indira IVF is the major name in India, which having the very much shareholding in India. After that, Nova is there. If you are, this is a whole India, there are IVF chains. If you are talking about the specific, which center in Jalandhar, so as I am working in this city, so I know the basic about which center is good. So in Maharashtra, like our center, Femcare Fertility, like Progenesis, they are the just state, they are the district-wise or state-wise center, they are good in their place. But overall, Indira IVF, Nova, Oasis, these are the good IVF chains.
So what was your total sample size?
Sorry, sir?
Total sample size.
It is 1046.
1046.
Yes, sir.
Okay, good.
Yes, thank you, sir.
Okay, very good. So, what is takeaway from out of your PhD program?
So, take off home is that's if we are working, this is as I already said, it's a social problem. This is not a personal problem. So, first of all, awareness should be there about the infertility. Second one is lifestyle. So, if we are seeing in metro city, his lifestyle is very smoking, alcohol, drinking. They are there, just weekend plans are there. So if we are working our lifestyle, so this problem may get overcome. This is a take-home message we can.
Do you think this problem will reduce in future?
So this is what I think, sir. So lifestyle.
I don't think, because with time, a lot of other things are increasing.
Yes.
So it means this problem will further increase.
But for that, maybe government should.
Government should.
Yeah, should do something for guidelines and all.
Guidelines.
Yes, sir.
Okay.
Even though I am just discussing with sir also, there is one major thing. For example, right now, if I was in 20, my 20 years, so at that time, if I frozen my semen sample, even though any girl who was in just 20, if she froze her eggs, so after 35, so that period there are some exposure I get during my whole life, which may affect my fertility potential. So instead of using sample of 35 years old man, we can use that frozen sample of which having the 20 years. So that may increase our fertility potential. So sperm freezing and oocyte freezing is also the trending right now.
Okay. I think from my side.
Okay, thank you, sir.
So should we declare?
Yes, definitely. So I congratulate to you for giving answers satisfactorily, and I recommend you for the award of PhD.
Thank you so much, sir. Thank you, ma'am. Thank you, sir. Thank you
Now
all
we
of
can call
you.
him Dr. Kale
Doctor.
Sudarshan.